Dear students, today we will discuss next generation sequencing. It is a very important topic as well as it is a very eye catching topic. It is very much in the theme as of now. Everyone is talking about NGS, NGS, NGS. Then what is NGS? What is next generation? As like everything get revolutionized. Like in your industry, there is a primary revolution, then it go to the secondary revolution, it is go to the tertiary revolution, and then now it is talking about the quaternary revolution. Like that, we have the common methods for the DNA sequencing. So means next generation sequencing is talk about the sequencing method and do not talk about anything else. It just talk about the sequencing method. And what we can sequence a DNA. It's a DNA sequencing. We can sequence a DNA. DNA first of all sequenced by using that two-dimensional crystallography in the year of 1970s. Then in the 1997 there is another method came, that is a Sanger's chain termination method, and there is another method, that is maximum Gilbert method. So they are the common method. Like if you want to sequence, you just take a sequence, break down this sequence and perform the two-dimensional chromatography, identify ATGC. So this is a common method. It takes too much of time, from sometime it is from 10 to 12 year. Like Human Genome Project completely took 13 years to get complete. Okay, so it is a very tedious process. So as the company, as the industries has been revolutionized, then your method is also. So in the year of 2005, sequencer has come forward and they took sequencing runs from 84 kb per unit to 1 gigabyte per run. So the frequency of the sequencing has increased. The time which was taking was too longer, it's get shortened. So जिस काम को करने में आपको year to year लग रहे थे, we reached to the days. So that's why it is next generation sequencing. So it is mentioned here, the rate of progress is stunning. As cost continue to come down, we are entering a period where we are going to be able to get the complete catalog of disease genes. This will allow us to look at thousands of people and see the difference among them. Like US or some developed countries, they are talking about the personalized medicine, as we are not talking about as of now, but with the advancement of NGS technology, we will one day we will surely talk about the personalized medicines. So what is the basic of next generation sequencing? In principle, the concept behind next generation technology is similar to Kepler electrophoresis sequencing. DNA polymerase catalyzes cut down the incorporation of fluorescently labeled deoxyribonucleotides, triphosphate DNTPs. It is DATPs, DTTP, it is dinitrotriphosphates, say ATPs, TTPs, CTPs and GTPs into a DNA template strand during sequential cycle of DNA synthesis. During each cycle at the point of incorporation, the nucleotide identify fluorophore excitation. The critical difference is that instead sequencing single DNA fragment, NGS extend this process across millions of fragments in a massively parallel one. What happened actually? Process is same. This is a sequence. We break down it in different parts. We took this part one by one, and we'll go for the sequence by using DNTPs. Okay. So what actually happened? What is the advancement? What is the advancement in NGS is that here we are going to one by one. If we have 10,000 sequence fragment in a DNA, then it will take, suppose it takes 1 day, so far it takes 10,000 days to complete the process, and then the analysis process. So it is a very tedious process. So, what NGSs do is it takes the whole sequence, hold the fragment in it together and perform the sequencing. So, that's make the process very easy. So, what are the steps for that? It is the workflow is contains the library preparation, cluster generation, sequencing and data analysis. So, in the next video we will talk about the each step one by one. Thank you.